Oh, yes, we're recording. That's absolutely brilliant. Well, good evening, everybody. This evening, we've got a really interesting talk lined up from Matthew Shepard, who runs the National Mite Recording Scheme for England. Matt, I know absolutely nothing about mites. I have been scrabbling around in my garden, and I'm pleased to say, managed to find some. No idea what they are, but I hope after this evening, I'll know a lot more. Anyway, without any further ado, I'll hand the floor over to you, Matt. Matt Shepard, the floor is yours. Thanks very much.
Well, thank you very much, Paul, and thank you, Alan, for your introduction too, and thank you all for coming along. You've come to the right place if you don't know anything at all about mites, although I will be taking things from the very basics, but I hope it will give everybody a bit of a grounding in how to identify mites, at least to family level, using the key that myself and Felicity Crotty have developed. This course, as so to speak, normally takes place over an entire day. I'm going to try and cram an awful lot of content, I'm afraid, into a relatively short space of time. So I'm very glad it's being recorded. It means that you should be able to come back and refer to some of the slides. Yes, this is all about mites. My name is Matthew Shepard. I'm, as well as being the national recorder for mites, I'm also the soil biology specialist for Natural England as well. And it's really how I got into mites. I realised there was a bit of a knowledge gap about mesofauna. There were plenty of soil microbial specialists and a few people interested in earthworms, but it seemed to be almost vanishingly small numbers of people interested in mesofauna about sort of 13 years ago when I started on this role. And so I decided that this was an area that I'd try and develop my own expertise in. And so this is kind of a bit of a personal journey through mesofauna that I myself have made as well, that I've tried to capture and present here. So let's see if I can actually move the slide on. First of all, I'm just going to quickly romp through a little bit on mite ecology, and then we'll get down to the brass tacks on mite identification. So in terms of mite ecology, well, it's useful to know what a mite is, and evolutionarily speaking, we can consider them as part of this broader group, the ecdysozoa, of animals that shed their exoskeletons to grow, and that will include things like nematodes even, and other, completely different sorts of phyla. Then they fall within the arthropods, jointed limbs, that's fairly obvious. Within that, they're within the Chelicerata, which means that they have pincer-like mouth parts instead of chewing mouth parts like insects have. And within the Chelicerata, they're in amongst the arachnids, and I've got a quick family tree of arachnids on the right-hand side there. We tend to think of arachnids as being sort of spiders, scorpions, and mites, but there's quite a lot of unusual arachnids out there as well, including vinegaroons and whip scorpions and palpigrades. However, most of them, sadly, we don't get in this country. We are, however, blessed with very many mite species. And I wish I knew more about spiders, but maybe I shall leave that for my retirement once I'm done with mites. Another way of looking at a mite is to consider what it is in terms of its characteristics, and I'm afraid it's easier in many respects to consider what it isn't. This is because mites have got such a lot of different variations of their body types. You can, with an insect, you can normally say, well, it's got 6 legs or something. That's fine. You could go on from there. But if you want to talk about a mite, well, it could have 2 legs, 2 pairs of legs, 3 pairs of legs, or 4 pairs of legs. Generally speaking, they normally have a 1-part body, but these can be subdivided and subsegmented in some cases. Generally speaking, what they don't have is more than 4 pairs of legs, or no legs at all. Generally, they don't have a body that's divided into separate sections, body tagmata. They don't have antennae ever, nor do they have compound eyes, nor do they ever have wings, although they sometimes have things that look like wings, which we'll come to. They never have giant pincer-like palps like a scorpion, and generally speaking, they don't have segmented abdomens or eyes on a single dorsal turret, which would be the characteristics, of course, of the Opiliones, the harvestmen. The mites are, however, pretty superlative creatures, and despite this very simple body form, they have held a few world records. The strongest animal in the world, the last time I checked, for its size, I should say, is a mite, and you can see why they might be strong. This one here on the right, Euzetes globosus, is pretty much a walking motorcycle helmet. It's extremely tough. It's full of muscle and a digestive tract mainly. And you can imagine that something that's so strongly built might be pretty strong in terms of its ability to lift and cling onto things. But the title goes to this creature in the electron micrograph at the bottom left, that's Arachnids longicetosus. And this creature they have tested, and they've discovered that effectively by hanging things off it, it can cling on to a piece of paper, even if you hang the equivalent of, and I'm scaling it up to human levels, for its size, it would be like us hanging on to 7.5 double-decker buses before we let go. So that's how strong the Arachnids longicetosus is. A very tough animal. We also have, I should say the fastest animals in the world. We seem to be missing the fastest animal. All right, well, it's a little anistid mite called Paracetamol longicetosus. Again, evolutionarily speaking, we can look at how mites are taxonomically classified as well. And they split into two broad groups. The mites themselves are a subclass, but they fit into two superorders, the Parasitiformes and the Acariformes. And when you see a diagram like this, you can often imagine that there was a sort of simple split at the very beginning and then they started to spread out. But in fact, actually, it's worth pointing out that the Parasitiformes didn't actually appear on the planet until the age of the dinosaurs, until the Mesozoic era. And the Acariformes, however, were there sort of in the middle of the Paleozoic era and the Devonian period. So I've got an armoured fish to remind me that that was when that happened. So these two lineages turned up a couple of 100 million years apart. And they're not actually that closely related. In fact, there's probably fairly good reason to believe that there's no reason to put the Parasitiformes and the Acariformes any closer together than the Acariformes and the spiders, or the vinegaroons even. Now, within these groups, we tend to kind of focus on four main groups, which were these ones. If you ever read any papers describing mite communities, they tend to identify things only as far as these groups. They'll say how many A-Stigmatina, how many Aurobatida, how many Prostigmata, and how many Mesostigmata you get. And we'll be looking in more detail about what these groups' characteristics are. But they tend to give the impression that these are at the same taxonomic level, and indeed, once upon a time, they were thought to be. But if we go back to that evolutionary or taxonomic diagram, you can see the Mesostigmata are an order of mites. Then the Aurobatida and the Prostigmata, also we're throwing the Endiostigmata, which people tend to ignore. Those are all suborders of mites. 3 of those, of course, within the Acariformes. And then we also have the A-Stigmatina, which are in fact a cohort of the Aurobatida. So they're at completely different taxonomic levels. Now, to some extent, it still makes sense to break them into these groups because functionally they're pretty similar. The Mesostigs tend to be predators. The Endiostigs and Aurobatida tend to be decomposers. The Prostigmata live everywhere and do everything. And the A-Stigmatina tend to be ruderal creatures and also decomposers as well. In terms of diversity within the UK, we don't really know. I'm still compiling every day the species list, trying to get rid of bad species from taxonomic papers and adding new species as they get discovered or turn up in the literature. In my list so far, I've got 2,049 species of mites. These include 448 of the Mesostigmata, which I showed you. Those are the predatory mites. We also have 671 Aurobatida. That's the more sort of crunchy brown-looking mites. But that 671 includes the A-Stigmatina, of which there are 300. That also includes, incidentally, the 300 within the 300 things like the scabies mites and feather mites. The 918 species of Prostigmata might sound like a daunting amount for people looking at soil like myself, but in fact that actually includes 222 known plant gall mites and 283 different species of water mites, and there's a number of marine mites in that group as well. So in terms of the soil groups, we've really only got about 1,000 or so mites that live within soil, which is the area that I'm particularly interested in exploring. However, this total list is likely to be an underestimation, and new species are turning up all the time. The creature at the bottom right there is Dorycrinosus splendens, which was sent to me in a sample from the south coast. It's probably been there forever. It's found on the continent as well, but this was the first time it had ever been found on mainland Britain, or in fact on anywhere in Britain, in fact. So yeah, the rather strange worm-like creature with 4 legs there is a plant parasitic mite. And up at the top, we've got a water mite as well, a close-up of its beady red eyes. Globally speaking, mites are probably vastly diverse. This is a paper by Descamps and et al. From 2006 that tried to estimate how many unknown species there were. This is based on the rates of discovery and the amount of effort that's gone into it. And they reckon that there are 25,000 described species of mites, but they suspect that this is only the very tip of the iceberg, with 97% of mite species remaining undescribed. So there's probably, according to his estimates, fairly close to 1 million species of mites on the planet. I think this is probably an overestimate. I think there are probably fewer, because I think they're making assumptions about the tropics. This is the kind of assumptions that they're making. This is a mite soil biodiversity index, which shows where the diversity of soil organisms generally is highest. And as you can see, they're picking out the tropics, and they're saying that the Sahara deserts are low, and you get less abundance, less diversity in the chilly northern and southern hemisphere. However, I'm not entirely sure I believe this. There's been other studies since that are showing things like mycorrhizal fungi actually begin to peak with their diversity in more temperate zones. And so we may actually find out that mites don't play by these rules. In terms of where mites live, they pretty much live everywhere. As I've said, there are marine mites, but we've also seen freshwater mites. There are terrestrial mites. There are mites that live up trees. There are mites that live on human faces and in birds' feathers. The top right picture there is Demodex folliculorum, which is the human face mite. And the middle pictures there are a selection of feather mites that are found on and in bird feathers. There's even an entire family, the Halorachnidae, which is the top left picture there, that lives entirely in the respiratory tracts of pinnipeds, of seals, which is pretty interesting. However, they are very abundant in soil, and this is why I came to tangle with them. They're normally more numerous than springtails in soil if you take a Tulgrim funnel extract, and I hope you've all been following the video that I showed you on, which I produced, which tells you how to do that if you didn't know already. Very simple to extract a large and baffling number of soil mesofauna from a very small amount of soil. Generally speaking, there can be 17,000 to 250,000 mites in every square metre in a piece of woodland, and that's compared to about 130,000 or so springtails, and that's based on monitoring data from our own monitoring programme. Like I said, there's some unusual habitats down there. There's the little sort of creature at the bottom right is actually a guinea pig fur mite that I found on one of my pet guinea pigs. Pretty interesting. So, oops, sorry, let's just see. Yeah. So what do they eat? Well, some of them are detritivores. In fact, most mites probably are detritivores in the soil. Most of the Orobatida and the A-Stigmata, many Prostigmata, and even some Meso-Stigmata feed on rotting material. But when we say rotting material, why is it rotting? It's rotting because it's got fungus and bacteria and other things living on it. So the mites are effectively eating a mixture of fungi, bacteria, and rotting material as well. Some of them chew up solid food, and some of them will actually drink from fungal hyphae. So there are different modes of feeding as well as different types of food being eaten. And you can tell the mode of feeding by looking at the size of the bottoms of these creatures, because if you're eating a lot of roughage, then you also have to pass out a fair amount of roughage as well. And creatures that eat roughage tend to have very large anal plates to allow the passage of the dung pellets. Whereas if you have a liquid diet, you have very, very small bottoms. And you'll be pleased to know that the reason that we don't have crazily itchy faces all the time, thanks to the Demodex mites, is because they have no bottoms at all. They literally only excrete liquid waste through their skins. So that's fortunate, because mite poo tends to be quite a strong allergen for many things. That's why scabies make your skin itch and why people are allergic to dust mites. In terms of feeding strategies, another group are predators. These generally include most of the Meso-Stigmata and also the Prostigmata, many of those. They can have different strategies in terms of capturing their prey. Some are cursorial hunters running down their prey, like the Anistid mite at the top right. Others are ambush predators. The snout mite in the mid-right side of the picture, that's another Prostigmata, will lasso its prey with sticky threads of silk that it produces and sort of lashes out at them. They can also dig little things like nematodes out of cracks and crevices by extending very, very long chelicerae. There's a turtle mite in the bottom left, and that thing coming out of the front that looks like a worm is actually its mouth parts. It's capable of extending these and withdrawing them entirely into its body in order to reach into crevices and pull out nematode worms to eat. And they can chew these things up using pincers, or they can suck the juices out by effectively digging needles in and sucking juices from their victims. Generally speaking, all of the Pro-Stigmata are suctorial feeders, whereas the Meso-Stigmata will chew up their prey with pincers. There are also herbivores that eat plants. There are, for instance, some external herbivores that chew away on the outside of plants. The red-legged earth mite up there on the top right, Halotidaeus destructor, which is a very dramatic name for a very tiny creature, but it can cause a lot of damage on the continent. It's not found in the UK yet, but it's found in Paris. I've seen it in huge numbers in Parisian grasslands, so it may well be on the way over here as climate change happens. We also have creatures that spin webs around themselves as they feed on the plants. These are Tetranicus mites. They're sometimes called red spider mites, which is a term that often gets applied to many other mites that run around and are red. And there are also ones that form galls. That strange worm-like creature with 4 legs I showed you earlier, it was Eriophyes tiliae, which forms these little needle galls on the surfaces of lime leaves. So they actually cause the leaf to form a little gall. It causes almost no damage or harm to the leaf at all, and they just remain within there, eating a minuscule amount of plant material. Not thought to do very much damage at all in most circumstances. But of course, mites are also associated with parasitism, and it seems to have evolved many times, independently in the Meso-Stigmata, the Pro-Stigmata, and the A-Stigmata, not really amongst the other Orobatids though. And you can see how this might happen if you're a predator roaming through the litter. Then you might easily move into nest debris, for instance. And if you're in a nest, well, you might take a little nibble on whoever's living in the nest. And from that, you might then begin to evolve into somebody that relies on taking a nibble on whoever is in the nest. And then once you're an external parasite, it's a short hop to becoming an internal parasite, just finding a more stable and cozy environment to live in. But likewise, a decomposer can go through a similar process of initially eating, slime left by slugs, and then eating the slime as it's on the slug, and then eating the slug itself. So you can see how these things evolve. Many creatures are actually benign commensals as well. The thermite I mentioned on the guinea pig causes no damage at all to the guinea pig that it lives on, sometimes in their thousands, and simply drinks the sebum that runs down the hairs of the guinea pigs. Some of them have got parasitic life stages, but then go on to be adults as well. So the velvet mites and allies, the Paracetamol, feed on arthropods as larvae, and then they become predators as adults. And we've got an Apiliones there, which has got quite a number of Paracetamol larvae feeding on its legs. You often see them around the joints in particular. Chiggers do much the same job to us. These are trombiculid larvae, and they can actually carry scrub typhus and other serious diseases as well. But the rash you can see on that person's leg is actually caused by hundreds of tiny mite larvae burrowing their heads into that person's skin. Not nice. But what these trombiculid do nicely is that you can use them to illustrate the mite life cycles. All mites lay eggs pretty much, even though, well, some of them don't actually get laid. I'll explain that later. But when these eggs hatch, they hatch into, well, effectively another egg. That deutovum is sometimes called a prolarva. It's another quiescent stage. So effectively, it's an egg that hatches into another egg that doesn't really move. Then when that deutovum or prolarva hatches, it becomes a larva, which then feeds, and as it grows, it will then shed its skin again to become a protonymph. Now, when it's a larva, it only has 6 legs. As a protonymph, it gains a pair of legs, and those extra pair of legs are carried through into the adult. So it starts having 8 legs the moment it becomes a nymph, but larvae only have 3. Deutanymphs are often a dispersal stage. So if you see a creature mite attached to another animal, most likely it's probably a deutanymph of that species, which means that the adult is likely to then sort of develop from that deutanymph when the animal that's travelling on has stopped travelling. And then, of course, there are the adults, and the adults are the only stage which are sexually mature, and it's only them that are capable of breeding or indeed parthenogenesis. In terms of life strategies, there are some mites that are ruderals, rather like weeds. These mature and breed very rapidly. As a result, they don't have much time to put on defense, so they are quite poorly defended, squashy creatures, or they have cheap defenses like long hairs, such as this Tyrophagus species on the right-hand side. They tend to have short lives, breed quickly, and then disperse rapidly. And these are all characteristics which mean that they're very good at taking advantage of temporary resources, sort of high abundance of temporary resources. And one resource, for instance, might be cheese. and cheese mites fall within this category. Tyrophagus there is actually the creature that the French deliberately infest their mimolette cheese with to create that rather strange mite-caused crust. I think it's probably banned in the UK because it's effectively still alive when you eat it. Some of them, however, have a different life strategy. They're called K strategists or C or S strategists. These tend to mature and breed more slowly. As a result, they tend to be well defended and they live long. So the creature there, Amauronophilus marinus, has been found to live 6 years in the Arctic, for instance, which is quite a long time for a creature the size of a full stop. It also has tough leathery armor. Other ones, as we've already seen, have got sort of hard, shiny armor as well. They tend to live a long time, but they also disperse very badly. They often the only dispersal mechanisms they have is walking. And they tend to be adapted to eke out a long life, growing slowly in stable environments with only slim pickings available. So a very different strategy. In terms of their dispersal, some of them can actually fly through the air on bits of just debris, or because they're very tiny. Some of them will send up a string of silk, which will enable them to balloon, like some spiders do. Red spider mites in particular are known to balloon. Other ones hitch lifts on creatures. I've got a sandhopper here, which has got some Phinocyrus fusicolus mites hitching a lift on it. But I've also got a close-up of these mites, and you can see that on the mites that are hitching a lift, there are some little white blobs. And those little white blobs are also mites that are hitching a lift on the hitchhikers. So that's called hyperphoresis, when you actually get hitchhiking of hitchhikers. But of course, there are some mites that spread around simply by walking, which is not terrifically effective, seeing some of them are extremely slow. This is a more efficient way of getting around. This is effectively it's a mite aircraft, effectively. We've got a Sexton Beetle here with a bunch of peaceful Acari Nymphs on it. And most of the time when the beetle is roaming around doing its business, the mites are hiding underneath its elytra, or hiding underneath various other crevices on the beetle. However, at some point they seem to realize that the beetle's about to take off, so they all run onto its back. They seem to align themselves, perhaps with the orange markings on the back, which is weird because they don't seem to have any eyes, but maybe those orange markings are to do with helping to camouflage the mites against others, who knows. And then when they seem to be ready, one of them apparently runs up to the front, that must be the pilot, and then the beetle is ready for takeoff with all of the passengers arranged neatly on the back. So absolutely lovely set of photographs there, which are taken by Jim Shaw and Richard Gresswell, I think, up in Shropshire. In terms of breeding, when breeding actually happens between male and female mites, it's almost always with spermatophores. There are some which do actually mate using an oedicus as well, but more often than not, the male will leave a spermatophore and then try to entice the female, or just leave it there for the female to find. In some cases, there is sperm transfer, where they will actually sort of grapple with each other and transfer the sperm using their chelicerae. In other cases, there's parthenogenesis, and the mite in the middle top there, which is Platynophilus peltifer, has actually been parthenogenetic as a species. It is reckoned for 100 million years. So this is before T-Rex walked the earth. This species existed, and it hasn't really had any sex since then. It has literally been breeding by reproducing itself, and it's now got 7 global clades, which are all genetically distinct, in some cases strongly genetically distinct, far more than any other normal species would be, but they still all look pretty much identical. And physogastry is another way in which they don't really lay eggs. The eggs are held within the mother's body, they hatch, and the young develop inside the mother. They even grow and shed their skins. In fact, they actually reach maturity inside their mother's body. Then the brothers and sisters mate with each other. The males at this stage, in their final stage, don't even have a head, so they're incapable of eating. And then the females burst out of their mother, the mother dies, and they go on off into the world to go and swell up like a balloon and produce their own babies in much the same way. So a very extreme form of parental care going on there. And there's also instances of mate guarding as well, where some mites will actually find a quiescent deutonymph of a female and lift it up and carry it around until it's ready to mate with them. In terms of defense, well, there are some mites that stack debris on their back. These Demeid nymphs in the top middle have got a kind of hat of crud that they carry around. It's a very cheap way of making a defense for yourself, but it's pretty heavy. Some of them use a kind of waxy layer on the outside that's quite tough for a creature to get through. Others just have long hairs, as we've seen with the Tyrophagus. Some of them actually carry their own shed skins on their back. So in the bottom right-hand side, we've got a Cepheus nymph, which each ring there with its little set of spikes on the outside is a former skin. So the first one will be the prolarva, the next one will be the larva, the next one will be the deutonymph, and this is the tritonymph, for instance. They can also be camouflaged. The creature in the bottom left, a Camisole Mite, has a kind of waxy coating that it encourages algae to grow on, and you end up in bits of dirt and so on. So effectively, it looks like, and it moves incredibly slowly. It looks like a walking piece of algae. They can also jump to some extent. Lots of Aurobatids jump, but the mite at the top right, Linopodes motatorius, is quite well known for jumping. You can, if you give it a little prod, or if it meets something with its crazily long front legs that it doesn't like, it can use the back legs, which are slightly swollen, to leap backwards. And many of them, as we've said, can also be armoured as well, like the armoured Aurobatids. Here's an example of an armoured Aurobatid. This is Stegonacarus magnus. And as the one on the right is it walking along. If it finds any danger that it doesn't like, it pulls in its legs, it folds down its head, and it becomes effectively an impregnable ball of armour. And most Aurobatids, one way or another, have got some form of defence that involves pulling legs into crevices like this. Now, I'm probably going to be teaching my grandmother to suck eggs here. I'm assuming you all know how to use microscopes, so I'm not going to go through the moving parts of the microscope. But just to say, if you followed my instructions, you should now have some creatures in a petri dish that we can begin to have a look at down a microscope. But you do need to sort them out and choose a specimen to pick out. So if you've got them in your petri dish, I would recommend moving around the little bits of soil you've got using a paintbrush that's had almost all of the bristles snipped off, or just I've got a little bristle from a shaving brush that I mounted on a bamboo skewer with a piece of insulating tape, and those are pretty good for just sorting through, because you're operating in a very tiny scale under a dissecting microscope. When you want to transfer a creature from your petri dish to a well slide, to a cavity slide, then if you use a small Pasteur pipette, something about like that, I think that's a 1.5 mil. Yeah, it's a 1 mil Pasteur pipette. But when you use it, don't squeeze the top, because that just sucks up way too much fluid and it starts blowing everything around the petri dish. If you just squeeze in the middle like that, that will actually enable you to suck up a tiny amount of alcohol with just the mite that you want. And you can be pretty accurate with that. You can normally pick out just one creature and get it onto a well slide. You can also sort together similar animals and also check your. But if you're doing that, it's worth checking that if you think they're all similar, that they really are, because some of them can look very similar under a dissecting scope, but in fact prove to be very different under a compound microscope. It's also worth switching around the base of your dissecting scope as well, looking at both dark and light backgrounds. If you only look at the light background, there may be some little white mites that you fail to spot and fail to recognize and pick out or count. So those are my top tips for manipulating mites under dissecting scopes. And when you have a compound microscope, again, I'm not going to tell you how to use it because I'm sure you all know. But in terms of I'm just going to start halfway down this slide. It's always worth using top lighting. I know it's a bit weird. Compound microscopes, annoyingly, mostly only come with lighting from underneath. And if you're looking at anything that is even slightly opaque, then it ends up looking pretty much like a shadow, unless you've got a phase contrast ring that you can somehow focus the light around it. However, top lighting is extremely useful. If you just get a bendy LED light, and I'll show you which one I favour, you can poke it just onto the stage of the microscope, and really it lights it up very, very well. So I'll show you the results of that later using the microscope I've got hooked up to this computer. Another thing that's worth doing is I tend to just put specimens in a well slide in the alcohol that they were collected in or stored in, and I'll identify them like that, and then I'll take them out still loose, and I will store them in more alcohol again. So I very, very rarely, if ever, make a permanent mount, which is probably something that you will think is utterly horrifying, and perhaps I should, but I find that I simply don't have the time. So I dare say, however, I mean, there are some great microscopists out there that love making beautiful, beautiful permanent mounts, and some of them are real works of art, so I will leave it to them perhaps. In terms of how you can manipulate a specimen, it is always worth being able to move a specimen around, see if you can roll it over, see if you can see both sides. The key, I'm afraid, does talk about features that are seen on both sides of the creature. So having sorted it out, if you can transfer one into the well on the well slide and drop the cavity slide over, inevitably you'll end up with a bubble, and you can normally get the bubbles out. Again, you've probably done this many a time, just by if you tip the slide to one side so the bubble floats up to the top, and then gently, very gently move the cover slip over, you should be able to release the bubble out of the side and top it up with a pipette. And then when you're done with it, you can either pick it up with a pipette again, or you can just wash it into a collection bowl, a collection dish, depending on what you intend to do with the creature that you've identified. Other top tips, I have once or twice myself managed to crush a mite into oblivion by failing to put it on the right side of a well slide, so always worth running your thumb over the top. Top lighting is your friend, and this light here is from, it's an IKEA light called Jansio. I can send you details of those if you like, but it's really good because you can angle it in many different directions. It's super bright, it's mains powered, it lasts a long time, pretty reliable, and you can fit it, the head, which is really small, underneath the objectives, or very close to the objectives of a microscope. If you need to move your creature, you can actually just move the cover slip left and right. That tends to reorient the creature. Sometimes it will roll over, sometimes it's too wide and flat to roll over, and you may need to take the cover slip off and then put the creature on again, having reorientated it on a dissecting microscope. And what I'm probably guilty of right now is letting my specimen dry out, because I've got a specimen on the microscope now and I've been talking for a little while, so they're probably drying out as we speak. But when you do that, you'll end up with a tiny little puddle of alcohol around the creature you're trying to identify, which will completely obscure all of its edge features and make it very difficult. So please don't let your specimen dry out. I normally tell students health and safety, if you're working with alcohol, then it is flammable and it's also poisonous if you're using IMS. So don't be silly and drink it or set fire to it. And of course, take breaks. Now, onto the key. This was developed to support the Field Studies Council course in soil mesofauna, because there wasn't really an accessible key to mites that didn't that went to a reasonable level, but also had any kind of illustrations at all. And to some extent, I started writing this key because I wanted to capture my own experiences, my own sort of top tips for how it was I was able to identify the mites. And so this has kind of become a key that's based on the shortcuts and features that I've ended up looking for, as well as some that I've lifted out of other keys. It does include a section on mite ecology, a section on collection, and a section on anatomy. It is a dichotomous key, which I do find to be the easiest to use, and it's not taxonomically strict in that bits of families end up sort of separated out in different parts of the key. I have, however, tried to colour code the main groups together, so all of the Aurobatids will at least lump out together, and all of the Pro-Stigmata will lump out together, and so on. Why haven't I published it yet? Well, it still needs more photos. It's illustrated with light micrographs, which I've taken myself using photo stacking software, but I still need more photos to illustrate it. So again, having a whole bunch of microscopists like yourselves out there collecting mites and taking photographs, and it would be really wonderful if we could include some of your photographs in the key. And there are some groups, I think, that I could probably still come up with better ways to key out. So it's still very much a kind of test version, and your feedback would be very welcome. Right, on with the identification part of the show. The basic body plan of a mite is that it only has one body tagma. That's compared to 3 tagmata in insects or 2 in spiders. It usually has 4 pairs of jointed legs, although in some groups there can be 3 or 2 or generally, oops, sorry, let's go back up, page up. There are normally 6 leg segments. Having said that, some may be fused, some may be lost, some may be subdivided. Mouth parts tend to be made out of pincers, chelicerae, and palps, which are leg-like appendages both sides of the chelicerae. There are often other features as well that are worth looking for. And all of these things tend to be adapted by fusion, development, and loss of body parts, which is why it's much easier to describe what a mite isn't than it is to describe what a mite is. There are various, we tend to number the legs 1, 2, 3, and 4 in Roman numerals. The head end of the mite is called the nathosoma, which literally just means the head, mouth body, region of the mouth parts. The region of the legs is called the podosoma, and you get so the propodosoma is the legs 1 and 2, and the episthodosoma is the legs 3 and 4. There's a kind of line that crops up quite a lot between legs 2 and legs 3. And on the back of the mite, that gets called the sejugal furrow. It's sometimes called the sejugal line, but often that's a line of division that seems to be important in mite anatomy. The rear part of the body is sometimes called the hysterosoma. When you're looking at the back of a mite, and often that's the most of the thing you can see. It gets referred to often as a notogaster. All of these are in the key in a glossary at the back. So I hope that you'll be able to understand. I've tried to limit the number of different jargon terms that we use in the key. But if I start talking about the notogaster and you can't remember what it is, then stick something in the chat and say, "What are you talking about? What's a notogaster? What's a propodosoma?" Okay. Also, mite legs are named. The articles of the legs, the joints of the legs are named. They're named in a similar kind of system as they are for other invertebrates. We have starting from this end, an apatele, then a tarsus, then a tibia, then a genu, then a femur, then a trochanter, never really known how to pronounce that, and then a coxa. Now, as I've said, some of these can be fused. The coxa on the left-hand side of the upper leg is short and ring-like. The coxa on the bottom here is actually an unusual shape, and that's because it's been fused into the middle of the body. So you can imagine other cocci arranged around it that are adjacent to it that are fused. At the end, there's an apatele, which may be a hook, or 3 hooks, or 2 hooks, or hooks and brushes, or just some brushes, or a sucker, or something else. Right, let's move on to the first main group, the order Meso-Stigmata. These are mostly active predators. There are some parasitic ones, such as the chicken red mite is a parasitic one, and some of them are kleptoparasitic, which means that they steal food from other organisms. The classic kleptoparasite is lives underneath the chins of ants, and it's called the Antennophorus, and it sits underneath the chin of the ant, stealing its food with its long legs as the ant eats. So seems like a fairly risky strategy. The order Meso-Stigmata are characterized primarily, I mean, again, there's many different shapes, but the fact that their leg cocci, from leg coccis 2 to 4, are shaped like short rings. If you can imagine cutting up a toilet roll into little segments, that's what the leg cocci would look like. They're like short rings of toilet roll. They never have eyes. Almost all of our UK species, not the one that lives under the ant's chin, but almost all of the other ones are in the group Monogynapsida. And there's a part of the key which asks for, it's a really awkward little question about the presence of a pair of very small setae on the back leg. And that part of the key is trying to determine whether you've got an unusual trigynaspid mite or a much more typical Monogynapsida mite. So if you find yourself squinting down the microscope looking for a pair of small setae on a little plate on the back leg, you could probably say, look for the common one, and it's probably down there. So it's not a bad place. If you're only starting, that's probably a good place to keep going. There are major divisions between the cohorts of the Monogynapsida. The Gammasina, which are ordinary looking predatory mites, such as the 3 on the right and the 1 on the left there. But also the Europodina, the turtle mites and allies, and we've got one turtle mite on this page, which has got it's the one second from the left on the bottom, just there. In terms of the order Meso-Stigmata, key features to look at, there is sexual dimorphism for starters. So on the underside, we've got the male on the left that's got one large sternogenital shield, and on the right, the female has got a genital shield and a sternal shield separate, often with metasternal shields either side. There's also an anal shield, or a ventrianal shield in this case. Another thing to look out for are these, as I mentioned, these leg cocci, which are always ring-like for legs 2 to 4. The ones at the front can be different shapes. They can be tubular or like doors even. But and I'll show you what that looks like in a bit. But these ones looking like little rings of toilet roll cut up is a really classic sign of a Meso-Stigmata. The other thing is most Meso-Stigmata have got peritremes. These are these grooves that run down the side of the mite, which are associated with breathing, and they have a little stigmata at the end. So if you've got a peritreme running into a stigmata, or a stigma at the end, stigmata is the plural, then you know you've got a Meso-Stigmata mite. And in fact, the name Meso-Stigmata means literally a hole in the middle of the side there, Meso-Stigmata. Another feature is the sternal shield and the genital shield completely cross over the longitudinal ventral line. So if you can imagine a line going all the way down, a sort of a dotted line going all the way down from the mouth parts down to the anus, the shields, the sternal shield or the genital shield, completely cross that line. There's no gateway in the middle. So they don't open up like Bombay doors. You do often get quite a lot of nymphs. This is a mesostig nymph. It hasn't photo stacked quite correctly because it looks like the photo moved there, but it's got the main features of a mesostig nymph. It's not very well sclerotized. You can see it's got a lot of soft white tissue. It's got a small sclerotized plate at the bottom, but that sclerotized plate doesn't have any genital openings. If we go back to the page before, the male has got a little lip at the front for a genital opening, or sometimes some males have even got a little circle in the middle for a genital opening. And the female here has got a genital opening at the front of its shield here, whereas the nymph has got no genital openings at all. It's not sexually mature, and it's got a lot of white space. If you see a lot of nymphs, and you probably will not be able to get them to species unless it's by inference to other groups. This family key tends to kind of abandon you when it comes to nymphs. Having said that, there are other keys published which do go to species, and they'll often key out the deuternymphs, the females, and the males as 3 separate groups. So it may be worth persisting, especially seeing as a lot of the creatures that you get phoretic mites on beetles and bees and so on are often in that deuternymph stage, and they're often pretty well described. Mesostigmata mouthparts are made of chelicerae. These are these pincers at the front. Either side of the chelicerae are the palps, and the palps have often got a little fork of sorts on the last segment called an apatele, palp tarsal apatele. They also have a pair of horns, and these come either side of the 2 chelicerae, and these are called corniculi, and these are thought to be useful in feeding. And there's also a kind of a brush in between them, and that can be quite elaborate, called an internal malar. So it's quite complicated looking mouth parts. Behind the corniculi, we've got a set of subgnathosomal setae, and the arrangement of these can be important for distinguishing the major groups. And then behind all of the mouth parts, there's something which is kind of attached in between the gnathosoma and the beginning of the sternal shield, and it's called a tritosternum, and it's kind of like a feathery little bristle that comes out. And again, probably thought to be something to do with feeding, possibly also mating. Not quite sure what it is. So that's the mouth parts of a mesostig, and you can see the 2 leg cocci coming out either side of the mouth parts. So in terms of trying to determine whether or not you've got a Gammasina mite or a turtle mite, and the obvious thing to do is say, does it look like a turtle? And that gives you some pretty good clues. But the real deciding factor is the arrangement of setae on the subgnathosoma, which is why I'm focusing on it. If the setae are in an L shape, or you could draw around them to make a triangle on each side, so that's 1 triangle on one side, 1 triangle on the other, then you know you've got a Gammasina mite. Whereas if your setae are more or less in a row, 1, 2, and 3, more or less in a straight line, then it's likely to be a Uropodina mite. Uropodina mite. And the reason I'm focusing so much on this is because some Uropodina don't look like turtles. Some of them are masquerading as Gammasina-like mites. So if you've got setae in a straight line like that, then you know that you're dealing with a turtle mite. But often that you know you're dealing with a turtle mite because it looks like that. It's really distinctive looking thing. Quite often they have got little holes that their little short stumpy legs can be pulled into. These are called pedofossae, and only turtle mites have got pedofossae. Not all turtle mites have them, but if you've got a creature with pedofossae like that, then it will be a turtle mite. It will be Uropodina. Also, in most turtle mites, you'll also get this, the front leg cocci 1, have become flattened and rectangular, and they close together really tightly to actually shut up the mouth parts. So the legs here are folded in at the front end, and they've become a pair of effectively doors that close over the mouth parts for protection. And all of this is adaptations for protection. So those door-like leg cocci are often a really good indicator that you've got a turtle mite. The pedofossae are an indicator that you've got a turtle mite. But if you don't have anything else to go on, you may have to try and look for those subgnathosomal setae. And if they're in a straight line, then you've also got something that's allied to the turtle mites as well. Right at the beginning of the mesostigmata, however, we need to rule out ticks because they do turn up in soil samples sometimes. And quite often, somebody who hasn't had much experience with mites will say, "What's that?" "Oh, it's a tick. You've got a tick there." No, it's a disgusting tick. Well, they're often not ticks, and the reason they're not ticks is because the mouth parts are very different. Ticks are also in the Parasitiformes, so they're related quite closely to the Meso-Stigmata, and this is why they look quite similar. They've even got sort of, fairly similar looking leg segments. They still haven't got those ring-like cocci though, they're a bit more fused into the body. But the really distinctive feature of a tick is that it has a bulb-like prong on the front called a hypostome, which kind of grows out of the mouth parts, and it's got loads of barbs on it, which means that when a tick jabs its hypostome into your skin, it's very difficult then to get it out. So that's how you really tell apart a tick. They also don't have peritones. They do have stigmata. You can see here these little dots just below the legs, either side of the hind legs. There's a pair of little dots. There's a stigmata. So that's one group done. I think it'd be quite nice to have a go at doing some of these practically soon. But perhaps if I run through all of the other features, then we can maybe see what it is that you've found. Order Sarcoptiformes, these are mostly decomposers, so we're moving away from the predatory Meso-Stigmata, and we split them into two suborders, the Orobatida, which are the armoured beetle mites and the dust mites, and also the Endiostigmata, which are considered to be evolutionarily primitive, well, they don't say that if they can hear you, segmented mites, and it says few and rare in soil and litter. Actually, sometimes they can be very common in soil and litter, so it's worth noticing them. So the Orobatida are often very popular because they have a beautiful variety of different shapes in their armoured body, all sorts of strange flanges and different textures on their armour. They're hugely diverse. It does actually contain the A-Stigmatina, which we'll treat separately. These ones, the non-A-Stigmatina Orobatids, are normally well sclerotized, and most of them, the really key feature for detecting an Orobatid is that the mouth parts are hidden under a kind of a hood. It's almost like the toe of a shoe. If your mouth parts are your toes, then the toe of your shoe, if you imagine having a shoe with no sole, it would cover over those mouth parts in much the same way as this hood covers over the front of the mouth parts of an Orobatid mite. So you might occasionally see a toe sticking out the end, but for the most part it's covered over by that hood. And you can see all of these different Orobatids have got that hood covering their mouth parts on the top. So from underneath you can see that hood again, but the mouth parts are held within a kind of chamber under the hood called a camerostome. Another key feature of Orobatids is that the leg cocci are not free. They have been fused together into a plate underneath. Now, to some extent, that could be more or less fused, but they are always forming, the main part of the sternal area of the beast is made out of fused leg cocci, and you can see these little lines in between them called apodemes, which show where the leg cocci are fused together. Quite often they will also have a line. If you look, remember that imaginary line I drew down the Meso-Stigmata, and I said that all of the shields crossed over that line, the genital shield and the ventrianal shield and so on. In the case of the Orobatids, there's actually a part where the genital shields follow that line. There's a break, and also the anal shields following that line. So if your genital shields follow the line and open out, like a pair of doors like that. They often get described as Bombay doors, and people have been wondering why, because what are Bombay doors? Is it like a Venetian blind? No, it's like the Bombay of an aircraft that opens up outwards and sideways like that to enable bombs to be dropped. Well, this enables the ovipositor to come out, or indeed a dung pellet to be dropped in much the same way. So they open out hinged on either side. Another feature that's worth pointing out is this thing sticking out the side, a pteromorph. Now, I've said that no mites have got wings, but the Orobatids have got things that can look like wings. These are protective structures that grow, sort of extensions of the notogaster, extensions of the back that grow out and around the side, and they can often pull their legs to some extent down the side of those. Surrounding the ventral shields and the genital shields and the anal shields is often a ventral shield as well, which can be more or less fused. So from the top, the Orobatid has got a prodorsum, as I said, that covers the mouth parts. It can have ridges or flaps or flanges on the top. All Orobatids have got this thing. It's a little, almost looks like eyes. There are little tiny holes in the side, and it's called a bothridium, and there's a seta that comes out of it, which is really distinctive. So that's a bothridial seta, and you can put them together, and that's called a trichobothrium, the whole apparatus of the pit with the bristle coming out of it. I mentioned we've got the procerosoma and the hysterosoma, and the notogaster is the back of the animal. And this is that subjugal line again. And the subjugal line, you can see effectively distinguishes almost a bit like a head and a body of the Aurobatid mite. Strictly speaking, not a head and a body. If you take an oblique view on the side of an Aurobatid mite, there's all manner of flanges and flaps that they can use for defending themselves. These are all designed so they can pull their legs into these spaces. So we've got the pteromorph coming down the side here. You might have a pedotectum, which you can tuck its first set of leg segments behind. It might have a sublamella and a tutorium around which it can wrap its other legs, which are these kind of flaps on the side here. It may have a prolamella, again, a little kind of ridge under which it can tuck the front segments of its legs. So really all of its front leg can fold along its podorsum like that. They might also have these lamellae on the top, which are big flaps, again, to protect legs. There may be cusps on the end, which can grow out into prongs, or they can grow out into flaps, often with lamella setae on the end or tucked to either side. There may be rostral setae at the end and interlamellae setae at the top. And you can see again that bothridium is a kind of sclerotized pit with the sensillus growing out of it. So there's lots of different features on the front of a mite. The translamella is the thing that joins together the two lamella and is present in some groups. Again, important to distinguish Aurobatids in some groups. With the Aurobatidae, we tend to split them into higher or lower Aurobatids. In the lower Aurobatids, the knee segment is very similar to the other leg segments. It's kind of like a series of tubes. Whereas in the higher Aurobatids, the knee segment is much smaller and the leg flexes more. And so to some extent, you can spot a higher Aurobatid if just to ask if the mite has got knees. We also have genital and anal plates, often very close together in the lower Aurobatids, and often flanked by separate adanal or agenital plates. Whereas in the higher Aurobatid, it's all been more fused together. The ventral plate completely surrounds the genital and anal plates, which are normally just a single pair. They may have soft tissue at the ventral sagittal line in the lower Aurobatids, but they're all welded together into a kind of solid box in the higher Aurobatids, so you never get that flexibility. The lower Aurobatids never have pteromorphs that they can pull their legs behind, whereas the higher Aurobatids can sometimes have quite extravagant pteromorphs. So just to give you a few pictures to show what I mean by that, we've got lower on the left and higher on the right. The lower Aurobatid here, the genu, is actually quite a big segment. It's even bigger than the adjacent tibia there. But on the right-hand side, we've got a higher Aurobatid, and you can see the genu is just a little bead, a little tiny segment that's much smaller and allows that leg to bend. And again, this is an adaptation to pulling its legs behind protective structures. The genital and anal shields on this lower Aurobatid on the left are very close together, practically touching each other. We've also got some extra shields down the side. We've got an agenital and adanal shield, which are actually fused together in this one. But in the higher Aurobatid, you can see that the genital shield at the front and the anal shield at the back are really well separated, and that's really important for identification. Now we've also got this flexible area of soft tissue in the lower Aurobatid, so that effectively means that the whole front end of the mite, from the front two legs and mouth parts onwards, can wiggle around. This is, however, not good for defense. It's a kind of chink in the armor, and in the higher Aurobatids, they've welded it together. They're effectively an inflexible box with no soft tissue in that area at all. So that's some of the features that the key uses to distinguish a really key distinction between the lower Aurobatids and the higher Aurobatids. It also talks about pteromorphs as well. And sometimes it says that it may have small flanges, which I don't even count as pteromorphs. They probably either used to be or were developing into pteromorphs, but it seems to me if you can't actually hide anything behind it, it doesn't really count. It's just a little flap. In other cases, the pteromorphs may be continuous with the edges of the animal. So here we've got pteromorphs that kind of run into the edge, and you can see that they kind of form part of a continuous line that runs into the edge of the animal. Alternatively, they can be lobe-like, which means that they kind of hang down from the edge of the animal, and often the knee of the last leg pokes out quite a long way behind where that pteromorph joins, whereas in the continuous pteromorphs, it's obvious that that knee can be pulled up behind the pteromorphs for protection. Now, when you do have a lobe-like pteromorph, they can be extended. They can be large and hinged, extending forwards. And in this case, they actually look like ears. They're often referred to auriculate pteromorphs, and that's might remind you of somebody. And on the alternative is they're lobed backwards, and they've got sort of dangling earlobes. so to speak. So these are extending backwards, lobed at their rear edges, extending backwards. So these again are important features in the key. Again, there are nymphs, and often Orobatid nymphs look very, very little like their adult counterparts. Often only the mouths and sometimes the legs are well sclerotized, and certainly not the genital plates. Sometimes the anal plates are sclerotized. Generally speaking, Orobatid nymphs are worth giving up on. They are not well described in many cases. There's a few old marvellous Victorian books that have described them, and which I would recommend looking at because they're fascinating. But you can still tell that it is an Orobatid and not an A-Stigmatina because it has these Trichobothria. You can see the little pits either side with the little bothridial seta, the cancellus, coming out. So definitely a baby Orobatid of some sort. Now moving on to the A-Stigmatina, the dust mites and allies. These tend to crop up less in soil, but they do in some cases, and I've got a few here. They're usually whitish, poorly sclerotized. The mouth parts are normally visible from above, unlike the Orobatids, which have got the toe of the shoe. They may have very long hairs or be completely bald. They never have trichobothria, and they have weird headless nymphs, and I'll show you a picture of those. So in terms of a diagram, some of the key features, as I've said, no trichobothria, mouth parts generally visible from above, and from the underside you can see often the only sclerotized bits of them are these lines between the leg cocci, these apodemes, and this is because they need to be able to attach their muscles to something solid so that they can then move. And they also tend to have this V-shaped genital suture as well. So dust mites and allies tend to have these strong apodemes. When they're babies, they travel around. I've mentioned that they can be hyperphoretic, traveling around on other travelers. So these little white dots are in fact due to nymphs of acarid A-Stigmata. And the reason they're able to hang on is because they have suckers. They're not parasitizing, these mites, and in fact they can't because they don't even have proper heads. The head in an A-Stigmatine hypopus is effectively a little stump with a pair of 4 bristles on it, and doesn't actually have any capability of eating anything. But what it does have is this set of rather beautiful suckers all around its anus, and it uses those to cling on very tight to its host and travel around with it. Onto the Endiostigmata. These are really have a mishmash of characters. They're sort of half Orobatid and half Prostigmata, and they're usually very, very tiny. They're often segmented. They may have eyes. They may also have trichobothria, like the Orobatids. They may have a naso, which is like a nose-like structure, or sometimes not. And they can have 1, 2, 3 claws, or none. In the key, they tend to crop out all over the place because of this mishmash of characters. However, one thing that unites them all is that they are pretty tiny and often easy to ignore. They often have quite extravagant looking setae. This Nanorchestid here has actually got setae that look like small shrubs growing all over it. Quite beautiful. It also has a little latch setae on the front with a kind of trigger. It's very strange. Final group, onto the prostigs. These are very diverse and common. They're all suctorial feeders, but there any similarity between the groups tends to end. There are predators, there are parasites, there are fungivores. They're mostly rather squashy looking creatures. They tend to be poorly sclerotized, but having said that, there are some very strong exceptions. They're quite often red, but they can also be white, green, black, less often yellow, orange, or brown. Generally speaking, they are broad-shouldered, bell pepper shaped, if you like, or strawberry shaped, and they often have quite sausagey legs, and there's often a gap between legs 2 and legs 3. So you've got 2 pairs of legs at the front and 2 pairs of legs at the back, and a big gap between them. There's often highly modified mouth parts because they're all adapted for suctorial feeding, and these are often quite hard to see. They also often have palps, which have got strange claws on as well. So this is a kind of typified Mesostigmata mite, and it's got the sausagey legs, it's got the broad shoulders, it has got eyes in this case. We've got some trichobothria. We've also got some strange peritones along the front. This is where they breathe, along here. We have paired claws. They nearly always have paired claws, often with an impodium as well, which can be a brush-like structure that sticks out. Oh, pretty interesting stuff. They have a genital plate with a longitudinal opening and a small anus because they are generally liquid feeders, so they don't need to poop out a lot. And they tend to have a gap between legs 2 and 3. There's that gap. So these are kind of the features of the whole group, but they've adapted it very greatly, as we'll see. At the front, there's a palp, and the palp sometimes has a claw. Now, the claw grows out of the tibia, which means that the tarsus, which is the final segment of the palp, is often adapted. So sometimes the tarsus will actually hang down like a thumb, and they call that a thumb-claw complex, because you can see how the thumb could be used as against the claw to grab things. And in other cases, the claw has got extra claws on the top, so I've got a bunch of families there that are associated with having these extra claws. In other cases, the tarsus itself has been shrunk, but the setae on the tarsus have grown out to provide something that they can use to grip against the claw. So the Cheiletidae, which are a Raphignathae group, have got these kind of bristles that it uses for grabbing its prey. In other cases, it's a bit simpler. The claw is literally just a series of spines, and the tarsus is pretty normal. And likewise, in the Erythreidae or the Smaragdidae, it's just a little spine on the side, and the tarsus is normal. So a variety of different thumb claws, all of which in the key will be described as being thumb claws, so being a claw on the palp tibia. So you do need to watch out for that. There's quite a variety of what we mean by claw. There are 5 main cohorts within that: the velvet mites, the Paracetamol, the Anistina, the whirligig mites, and other allies. The Raphignathae, there's an extra H in there, it shouldn't be, it should just be R-A-P-H. Raphignathae, which are stigmaid mites, also the plant-eating spider mites as well. Eupodina, earth mites, snout mites and allies. Heterostigmatina, which are often tiny in plants and soil, and other weird oddities like the Arnie mite. And I actually found one of these when I was making that video for you. And this is one of these rather well sclerotized mites. It's got kind of tough, bright yellow armor. And we call it the Arnie mite because it appears to have a 6-pack. Also quite fond of doing a kind of hero pose as well. So Paracetamol velvet mites, these are pretty familiar, and they all have a claw on the palp tibia, sometimes with extra spines, sometimes without. The shape of the setae is important in distinguishing these ones. The eyes are worth looking at because if they are stalked or sessile, that's important, and that whether it's got 2 or 1 eye on each stalk or on each lump is also important. And the length of the legs compared to the body as well. Many of them have got short, stumpy legs. Some of them have got quite long legs. The Anistina, whirligig mites, there aren't many. We only really have a handful of genera in this. And these are weird because the legs at the back are close to the legs at the front. They all branch out from a sort of single tuft on each side, presumably adapted for running really, really fast, which of course is what they do, charging around on plants and on hot stones in the summertime, chasing down their prey. The Raphignathae are a really mixed bag of mites. There are plant-feeding mites, soil mites. They also have a claw at the front of their palp tarsus as well, often with a little thumb hanging off it. And the arrangement of the empodia on the claws are often adapted or have sticky hairs coming off them, and sometimes they have odd features at the front. This one at the top right, which is Cryptognathus Lagina, has got a conservatory effectively on its nose, which it withdraws its mouth parts into, and it's one of 2 unrelated prostigmatas that have conservatories on their noses. So quite bizarre. The Eupodina snout mites and earth mites and other allies include lots of predators and a few fungivores as well. These will include the slug mites that you often see as well. Snout mites break the mold by being not particularly broad-shouldered. I suppose they still have a broad shoulder, but they also have a tapering nose. We also have the earth mites, the Linopodes and Eupodes, which have got strong, bulgy back legs, and very fast-running raggediad mites as well. And then finally, the very weird-looking soil-dwelling Heterostigmatina. These include tarsonemid mites, which can also be parasitic on things like bees. That's the bottom left. They're called tarsonemmids because their back legs look like little worms. That's what that means, tarsonemid. The top left is a pygmaphorid mite. That's a soil dweller, and we've already told you how they breed. And we also have the very bizarre-looking, it looks a little bit like a horseshoe crab. That's a scooter-carried mite. And it's got very long bristles on its back legs. So really, really distinctive looking creatures. And now I think I'm going to stop there because I've been talking for the best part of an hour. And I think it's probably time that we sort of throw it open to the floor and find out how you got on with your collections, and if you have anything that you would like to share and perhaps apply some of the the features that I've been talking about. So thank you all very much. And I've got a few slides, to wrap up with, but it would be quite nice to see what people have found at this stage. I think make it a bit more interactive.
Okay, Matthew, thank you so much. That was absolutely fascinating, and I had absolutely no idea that there were so many different orders or families or groups, however you're subdividing them, of mites. I'm in awe at your fluency.
It's a whole world. This is the thing: is when I found springtails, I started with Mesofauna. I started with springtails because there was an available key, and also because to some extent you only have to learn a small number of moving parts. Whereas with mites, it's an entire world. It really is. It throws it right open, and there's such a lot of variety. And to some extent, if you get your eye in, it becomes easier, because after a while, a lot of springtails, you end up sort of looking for features of CT on hair, hairs on hairs on hairs sometimes. And with mites, however, you can often just look at it and say, well, that's obviously a such and such, but the difficulty is trying to categorize them in a way that actually makes any kind of sense, because they're all so different from each other.
Well, it's absolutely fascinating. Now, just a very quick word. I'm going to ask Alan something. Alan, do you think we should stop the recording at this point? Have you got a limit on how long you can make?
We haven't, but I'm happy to stop. I actually had a couple of questions for Matthew that might be useful to have on the recording.
Fantastic.
Could I do a screen share briefly? Matthew already mentioned the famous key. This is the version I've got. I don't know if this is the latest version.
It is actually, I'm afraid. I haven't updated it because we haven't had another FSC course. I normally update it a little bit every year, but I'm afraid with COVID, the course hasn't run. So I've been working on other keys.
So you kindly distribute this via the Facebook Soil Organisms group. Would we be able to make that available to people at this seminar?
Yes, we can. I can paste a link into the chat. That might be one way.
Yeah, that's great. If you could do that.
Or if somebody else has got it, they might be able to paste a link into the chat.
Okay. And I've been charged to ask you a question. I'm asking for a friend, quite literally, about the recording scheme. Does the recording scheme include the plant gall mites where the mite is never seen, but only the gall is seen?
The vast majority of records for Acari are plant gall mites, because it's actually easy to identify them, and they tend to be pretty faithful to the plants. I'm not actually any kind of an expert on plant gall mites, and that's partly again out of necessity, in that there were already quite an active plant gall.
Well, that's what I was going to say. That is that there is a very active plant gall community. I imagine the vast majority of records going into the recording scheme would come from the plant gall community.
Exactly. And the thing is, I've set myself up as on iRecord as being a verifier for mites because of the soil mites and creatures that I'm interested in. And of course, it keeps pestering me, saying, "Oh, there are more plant gall mites for you to verify." I don't know anything about these.
We help you with that in Leicestershire and Rutland.
I did contact some time ago with the Plant Gall Society saying I'm setting up a mite recording scheme to record things that aren't plant galls, and would you be interested in sort of overlapping records and sharing resources and so on? And to be honest, I don't think I've heard very much back from them, if anything at all. So maybe that. I mean, I'm quite happy to go on to the NBN and onto iRecord and pick up plant gall records, because there's no harm in having the same data in two places. It doesn't make the mite seem excessively common.
That was my next question. What's your favoured method of people getting records to you? Is it via iRecord?
Well, iRecord would be great, but the problem is the taxonomic listings on iRecord aren't. They don't even reflect the list that I've managed to assemble, and this is a problem because we have a problem in that the taxonomy needs updating. And thanks, Lukey. You've just posted a group chat which says the Facebook group has got it's basically the link to the key. So sorry, yeah. The problem is that iRecord has to run off the national species list, which are run out of the Biological Recording Centre run by Chris Raper in Natural History Museum, and I've been working with Chris to try and get the mites updated, but he's snowed under with many other groups as well. Mm-hmm. And to be honest, I keep changing the species list because I keep finding publications that ends up, reducing the number of species here and adding more species there. So it's a very fluid thing, and to some extent it's quite difficult to get those kind of lists ready for people to choose from a drop-down menu on iRecord.
So what's your favourite method for people to get records to you then?
Well, if it's on iRecord, then please put it on there, because I will be able to get it.
Okay.
And I will also be able to verify it. I mean, it depends on. I might be able to verify if the photos are good enough, as you've seen the size of some of these creatures is very, very tiny, and the features we'll be looking for are often very tiny as well, to get it down to species at least. So my verification may not be able to go all the way down to species, which is why it's useful to have groups like this. But I mean, the other thing is just to actually, I love it when people send me spreadsheets with like their annual catch or their annual observations or things like that, because anything that can be transferred rapidly into a sort of database form is ideal for me. Whereas anything that needs to be sort of harvested slowly, such as I do get records from the Facebook page, for instance, because we do actually have quite long debates on what certain things are. But I think maybe sending me spreadsheets to my email, and there is an email. Again, I could probably share it here, which is acari.uk at gmail.com.
Okay, can I briefly share my homework as well while we're here?
Yeah, go on.
So I've tried to use the key for the first time at the weekend. Battled with the terminology, of course. Confidently got this one to family. It was a pretty easy one, I have to say. So I think the key stops at family level.
It does. This is I suppose to make it not too off-putting for people. And the other thing is if we did go down to species level, then we'd end up with a key that was monstrously thick and potentially unaffordable. The good news is there are keys out there that are available for some of the groups, but not all of the groups. Yeah. Well, this particular group, the Trumbididae, the best key is it's this one here. Actually, I'm going to show it to you. It's I don't know where you could actually buy it. It's Trumbididae. It's in English, but it's the Trumbididae of Poland by Joanna Makol. Mm-hmm. And I actually got it by writing to Joanna herself with the query about a Trumbidiid, and she said, "Oh, I'll send you a free copy of the book." So I was like, "Thank you so much." Is it a signed copy?
So what the key does say is to take these further, it would need details of the setae.
Yeah. Now, what I've been doing for this particular group, because we get a lot of records coming in, a lot of people sending in observations to iRecord, and a lot of posts on the Facebook group as well. And the thing is, these creatures are very obvious, and there's one well-known species, Trumbidium Holocyrosaurum, and so everybody says if it's red and fuzzy, it must be that, and it's not sadly the case. We have many, many species of red fuzzy mites. And the problem is some of them we may not even have descriptions of as adults. We've only got red fuzzy mites that we've seen parasitizing other insects, for instance, as larval forms, and they've had a species erected, valid species, on the basis of the larvae alone. So we don't even know what they grow up into. Mm-hmm. So that's a bit of an issue. But because this group is so popular and people do like them, I've been actually coming up with another key along the same lines as the mite key, which brings all of the velvet mites to species as best I can. And at some point, I would really like to be able to trial that with people and see how they get on with it.
So if I wanted to try and take this one further, what kind of magnification would I need on the setae?
I reckon about 400 times. When I say that, I mean a times 40 objective and a times 10 eyepiece.
Okay,
will do the job.
And do you do that on an intact specimen, or do you fragment it some way, or? Yeah,
no, I do it on an intact specimen. And again, this is where top lighting comes in, because if you have a creature like this, it's often very difficult with a compound microscope to see any detail in the features when you've got the light shining from below. Whereas when you have light shining from above, and these are quite big creatures, you may be able to see quite a lot of details with 100 times magnification. But even at times 40 magnification, you can still see a certain amount of detail. I don't know if it's worth my while sharing my screen with this with my microscope software, just so that people can see how detailed an image you can get. I have let my specimen dry out though. So
okay. Well, just before you do, my other piece of homework, which is another easy one, and again, I got as far as the family from the key, which very nicely describes the 8 setae in 2 rows.
Right.
I could do that. That was fairly easy. But again, I think, am I correct in saying the key stops at family level for these?
The key stops at family level for the Domeidae, yes. This is a really diverse group, sadly. So, and again, big and obvious. So we get lots of people saying, oh, well, what's this? And again, there's a relatively common one, Domaeus onustus, which is mentioned in several books. And so everyone assumes that they're all Domaeus onustus. To get this one to species, there's various things you need to look at. You need to look at the setae on the leg segments, and count them up and work out whether you have certain sort of paired setae or not. That can in itself can be a bit of a pain. So again, you'd be looking at trying to get 400 times magnification. And to be honest, you end up often either having to photograph it from many different angles or draw little temporary diagrams to work out where the setae are. And another thing that's a really important feature that all the books talk about are to do with little lumps and nodules, which are found sort of in the crevices, sort of between the body, the notogaster, and the prodorsum. And you can see that with this creature, it's not the cleanest. They don't have the cleanest of habits. They often wear hats made of crud when they're in their nymphal stages. And this, which I think is an adult, is still covered in quite a lot of wax and crud and algae and fungus and all sorts of stuff. So but actually being able to see the lumps and bumps that you need to get it to species, it can be really quite challenging. I find these some of the most frustrating ones to get to species. And what I'm hoping is eventually I will be so familiar with them that I will be able to work out an easier way that's based on broader features. Because like I said, with a lot of these, a lot of the keys that you get are based highly on strongly taxonomic justifications, and the taxonomic justifications are often raised by saying, well, we know that this seta has evolved in this particular way, so we'll only base our key on this particular seta because it's a really good way of distinguishing it. Now, often it might not be the most obvious thing. You might end up with 2 different species, one of which is bright green and the other one is bright red, but the key will still be talking about that blasted setae. And it's
so what you're saying is that the teeny tiny ones might in fact be easier than the rather large and obvious ones.
Yeah, I mean, it depends what you can get hold of. There are many free identification resources, often ones that can be found on Biodiversity Library.org, which are is a fantastic resource. You go back, you can search by species, you can search by author, you can search by, and it helps if you know a little bit about what the species used to be called, because often these publications are from the 1950s, 1940s, in some cases even the 1800s. We have some resources from there, but often the descriptions are still good and the species are still good, and as far as we know, there hasn't been anything that's been published on the British fauna that's superseded it. So in many cases, it's worth going back to these old resources. And increasingly, what I'd love to do is to produce a key that updated all of these. But in many cases, I mean, here's a grand example. This is relatively recent. I think this was from 1980. I don't know if you can see that. No. But this is a mites of the subfamily Paracetini, completely free. I've printed it out at work. But it takes all of the one major subfamily of the Paracetini, to species, and it's got all the deuternymphs illustrated as well, and it's it is out there. And the thing is, what I'm hoping to do is I've got a website that on Scratchpad that has got all of my species list on, but at each stage where we've got a resource like this, I try and put a link or the actual file. If it's freely available and I can download it, I'll actually put the file on that as well, so that all of these resources are gradually piling up in one place. And again, it's kind of like an ongoing work. So whenever I find a new piece of ID, then I'm able to upload it to or even just upload a link, just so that people know where they can start looking.
So can you share the link for that?
For that particular one?
Yeah, for your site with all of your.
Yeah, that's I'll see if I can. I'll probably have to go to it and then copy the link, so I hope it doesn't crash because I'm having to open yet another piece of software. I was going to show you the joys of top lighting, wasn't I?
Yeah.
So let's see if I can get. I'm just going to. Right, so what we have here, let's see. I'm going to share my screen again. I can just share that. So this is a turtle mite, as you can probably tell, but this is just with the lighting from underneath. And as you can see certain features fairly well, but where you get to the middle dense part, it becomes very dark and quite hard to see what's going on. And likewise, if we get to an even higher magnification, it's not too bad. And the thing is, actually, in some cases, it looks better on the software than it does on the down the microscope itself. Because I think there's kind of a correction factor going on, so you can see some of the CT there. However, with top lighting, I just get my bendy light and poke that down here. We get a rather different view. So it's a bit overexposed, but there you can actually see the colours.
Yeah,
it's taking a while to update, and a bit more of the individual features that you get. And even with the ×40 objective, so this has only got like a sliver, about less than a millimetre between the objective and the cover slip, and yet that bright LED light is still able to shine in there and show quite a lot of the features on the surface there. Yeah. So it is very much worth getting a good strong top light that you can use to illuminate the features. And it does, most of the old mite books as well will tell you that what you need to do to identify anything is to soak it in lactic acid on a hot plate for a few hours and then mount it. And the problem with doing that is A, it's a hassle, B, not everybody can get hold of lactic acid, not everybody has a hot plate, and it's also to some extent gets rid of certain features as well. I mean, it might clear up your Domaid mite, but it will probably remove the colour, for instance, from it. And while it might make some things easier to see, and I think it probably would be easier counting leg CT on a cleared specimen, sometimes they literally just never clear. They're so well sclerotized you could spend hours trying to clear that Domaid mite and it would probably still look pretty inky black. So I think this is why they're seen as being challenging, whereas just now that we have really bright, cheap LED lights that you can just poke under the microscope, under the objective lens, then you can see it does a tremendous job of revealing some of these features. So here I think you can actually see that line of 3 CT in a row that distinguishes the Aurobatids here. Yeah, there's a CT at the front. So if I can point it, I don't know if you can see my CT there.
Yeah.
CT there and a CT there. So that's 3 subgnathosomal CT in a row. It's not often that the doors open like that, so sometimes it doesn't do that.
Okay, thanks a lot. I think I probably...
Right, thank you. So I thought I'd mention a bit about the mite recording scheme. This was a scheme that I started up in May 2015. It actually contains, it says 9,620 records. It's now over, I think it's closer to 11,000 records. And the majority of them have been harvested from literature, from observations. I even get them off Facebook. They do include some, rather a lot of plant wool mites, but increasingly I'm trying to swamp those with soil mites as well. But there are many more from the countryside, from the iRecord and other sources, and presumably from the Plant Gall Society as well. There's also a very large chunk of Aurobatids mites that came from the Countryside Survey, which was some identification carried out by Frank Monson at Liverpool World Museum. And he did a tremendous job, went through all of the, I think it's about 500 soil samples, looking at all of the Aurobatids and identifying them all to species. So there's a tremendous amount of information associated with different habitats across the whole country. So Aurobatids actually are among the better recorded groups. It's good to gather all the usual recording information. Unfortunately, we can't get everything to species, but at the moment, because I just want to encourage more recording, I'm very happy to take even a cohort level record, just saying there was a Paracetamol, there was a velvet mite here. That's fine. I'm very happy with that. However, we also try to record microhabitat, because sometimes just saying, well, this is a mite that we saw in an oak woodland isn't very helpful. And so I created a system called the Shades system. And if you download the key, you'll find that in the back of it there is also the shade system. It's a way of describing in a very short code the microhabitats that you find in terms of the substrate, and you can have two substrates in case it's on the border between two. Estimates the humidity, the acidity or alkalinity, and the level of disturbance. And those are, probably the most important things when you're very tiny. You need to know what substrate you're on, how damp it is, how acid it is, and how long the situation has been there, how likely you are to have had to blow in on the wind. And you can actually analyze the data that comes out of this recording scheme to actually say, well, what situations are associated with what species? Because we don't really have that kind of horticological information about mites yet. So this is a way of trying to gather it as the recording comes in. So just looking at substrate, a lot of the effort has come from soil cores and the identifications by Frank Monson when it comes to species level ID. So we get lots of soil and lots of records. Where we have at least 20 to 34 records for a species, I was beginning to analyse this data. You can see that some of them do just crop up mostly to do with soil, like Mediopia subpectinata. That is pretty much a soil-dwelling creature. It's only really ever found in soil, but also in rotting organic matter. Well, not too surprising there. There are other things like Carabodes marginatus, which are more often found in lichen or organic matter, or in some cases on rocks. There's one over here, Fallopia Lucorum, that you can see is almost never found in soil and is only found in lichen, moss, and rocks. So we're seeing different habitats cropping up. And you can then begin to actually check this. So this is Fallopia Lucorum. And again, I went back to my Bible of Gerd Weigmann's Orobatida, and he says of Fallopia Lucorum that, and this is, I had to translate this on Google Translate, frequently found in moss and lichen coverings on trees and on stones, rocks, etc. So actually, the data that we're collecting here is backing up where we already have got some descriptions of habitat, but it's specific to the UK, which is really nice to be able to get that information. We can also look at disturbance means and ranges as well. So we have a kind of a sliding scale that's semi-logarithmic of saying, has it been disturbed within the last week, within the last month, within the last year, within the last decade, century, or pretty much undisturbed. And you can see that the species that crop up, like Malacanophilus monodactylus, is only found in pretty much completely undisturbed habitats. And then you get your cheese mite, Tyrophagus, which is found in everything that's been disturbed from the last decade to within the last week. So you begin to see that actually you can use these mites as a pretty good indicator of how recently something has been, the situation has been there. And in fact, this is put to good use in some situations by forensic acarologists who use it to determine how long dead bodies have been left in woodlands. There's somebody in northern Spain who made a whole career out of leaving pigs in woodlands and then studying the mites that appear to decompose their bodies. Quite grisly. Also humidity ranges as well. You might find some mites such as Nanomania coronata, which is that strange eye-browed mite over there, and that is only really found in the damper end of soil, whereas at the other end you've got our old friend Fallopia lucorum, which was found in lichen and moss and rocks, and as you might expect, it's found generally in dry habitats. And again, this just a kind of sliding scale of would this dry out in a day, or is it periodically waterlogged or permanently waterlogged at the very top? So it's a kind of a general easy to estimate scale of the hydrological regime, I suppose. However, we don't actually have that much mite data. If you go to the NBN and say, what have we got for Orobatida? This was taken from a couple of years ago, but there's not a lot on there. And the problem for this is simply because we just don't have reliable species lists. As I've already mentioned, the lists that are associated with iRecord are incomplete and out of date, and I'm working at the moment with Chris Waper to try to update these and improve the situation. At the moment, I've got all of the records held on Google Docs, and it's I'm able to actually do a kind of simple mapping effort by just doing a lookup table and plotting out the records as they come on a map. And we're beginning to work out that, yes, we've got some which are pretty common. Opiella nova turns up pretty much everywhere. There are other rarities which seem to be geographically restricted. Conopia palmicinctum is pretty much only found in the southwest of England. There's one record up near Arnside in Lancashire, but I don't trust it, quite frankly. I want to see that that record because I don't think it's correct. And then you've got other ones like Trimalacanothra sculptus, which are associated with boggy ground and only appears so far in Cumbria and the west of Scotland and north of Scotland. So definitely some habitat associations and some climatic potentially associations, or maybe actually to do with past glaciation and the fact that Calliope palmicington may have survived as a Lusitanian species in the unglaciated areas of Britain. Might also be found potentially in Southwest Ireland in that case. We do need more records, so if you can help me out, that would be fantastic. I'm always trying to do things like this to improve taxonomic expertise. We do try to run a regular training course at the FSC. This is an example of an ad hoc one. I've said ESB, that's the Earthworm Society of Britain. I should like to kind of model some of my work on what they do, but I am involved with them as well. We also have the soil biology group with the AAB, which is no longer active, sadly. I ceased to be the chair of that, and then it kind of stopped doing things. However, I am trying to develop better ID resources. It would be lovely to get some help in terms of sending photographs, or even in being able to say, here's this group. There isn't a good key, but I know the resources are out there. Does anyone feel like putting their hand up and helping out by developing a small key for this lot? That would be fantastic. And but ultimately, I think it's pretty accessible, this stuff. You don't have to have much in the way of specialist equipment, and microscopes that you can use to look at these creatures are not actually that expensive when you compare to what a birder will spend on binoculars. Microscopes are actually extremely cheap, and there are all sorts out there. So if you know of a source of good cheap microscopic equipment, and I'm sure there's much discussion as to whether the cheap stuff is any good on this forum, then that would be fantastic. And this is just a few resources. The Facebook group, if you're not already joined up with this, it's just a good place for posting things that you've seen, you want me to identify, or sharing events or ideas or observations or bits of news or anything. So please do join up with that. I mentioned the Biodiversity Heritage Library. This is the source of a lot of old published keys on also species descriptions, sometimes in Latin, sadly, but you can often do a bit of detective work digging around and find some really useful keys on this site. If you need to check more up-to-date taxonomy, then the Global Biodiversity Information Facility is pretty good. I'm not entirely sure where it gets all its information from. Sometimes I think, again, for mites, it's not 100% accurate, but it's normally pretty good to see what the synonyms are for various things. Increasingly, the BOLD database is picking up barcodes for these barcodes for individual species, and we'll be using these in monitoring and attempting to actually build up a barcode database. There is an ongoing effort in the UK to actually build up a kind of genetic centre of excellence that will attempt to barcode all UK species at some point, which is a really noble aim, but it would be extremely useful to be able to identify this stuff without having to gaze down a microscope. Having said that, it's really important that we do keep gazing down microscopes, because that way we discover more about the ecology of the species, and actually you can't popularize a barcode either. You actually have to show people the creatures themselves. This is the Scratchpad webpage, which now also has springtails on as well. It's got big photographs, species lists, and links to literature. So this is I'll type the link into the chat, but it's soilbiodiversityuk.myspecies.info is the one to go for there. And there are other resources as well, which I can probably point you to, but if you contact me on the Facebook group or directly at Matthew. The email I've already shared, that's acari.uk@gmail.com, then it would be wonderful to hear from you about the other resources you find too. Thank you very much.
Well, thank you very much, Matthew. That's a great talk. I think that's the most comprehensive talk we've ever had to this group. Thank you for giving up your evening. I hope, like you, I'm sure, I hope you've inspired a few people. I think we've gathered a more expert audience this evening rather than a casual audience. But I hope I find with these more difficult taxa, it's getting started, it's finding a way in, which is so often the problem. And certainly the key, and I think seeing things and hearing you talk through that this evening will hopefully inspire a few people to get started and plunge in a bit deeper. So thank you very much.